Hello guys, welcome to Dr. Science. So today we are going to talk about the biosynthesis of triacylglycerols, and this is the pathway that is taken from your Harper's biochemistry textbook, and this diagram is literally very tough to understand, but I will try my level best in to make it as simple as possible. But please watch this video in chronological order. So in my first video I talked about the overview, how the phospholipids are synthesized and how the phosphatidate can synthesize your triacylglycerols. And in my video I already talked how triose phosphates such as your glycerol 3 phosphates are synthesized from your glycolysis and lipolysis pathway. And in this video we will mainly talk about your triacylglycerol synthesis. As you can see here, we are going to understand this complete pathway. Okay, so first I will talk about the basics. In my basics I will be mainly talking about the triose phosphate, which is your glycerol 3 phosphate. Okay, now I already told you glycerol is a 3 carbon structure, and to this 3 carbon structure there is a phosphate present on your third carbon, hence it is called as your glycerol 3 phosphate. Now how this glycerol 3 phosphate is synthesized, I already told you. Either it can be synthesized from direct glycerol, it can be synthesized from direct glycerol, okay, by the enzyme glycerol 3 kinase. The enzyme is glycerol kinase. So glycerol kinase can convert the glycerol into glycerol 3 phosphate. And the other pathway is your glycolysis pathway, where you can convert the glucose directly into your glycerol 3 phosphate. And finally we have another pathway, which is your where we can convert dihydroacetone phosphate into glycerol 3 phosphate, and the enzyme responsible here is your glycerol 3 phosphate dehydrogenase. Okay, so we can convert the glycerol, glucose and dihydroacetone phosphate into glycerol 3 phosphate. So as you can see here, this is the glycerol 3 phosphate, which is a type of triose phosphate. And from this triose phosphate we are going to do a series of acylations. And from this series of acylations we are going to form your phosphatidate, and from phosphatidate we are going to synthesize your diacylglycerol and finally your triacylglycerol. So we have to understand this entire pathway in the synthesis of your triacylglycerols. Okay, so in the synthesis of triacylglycerols we are going to convert the glycerol 3 phosphate into phosphatidate and finally into your diacylglycerol and finally into your triacylglycerols. So this entire pathway we need to understand this one in a crystal clear manner. Okay, so I'm going to draw. In order to understand this, I want you guys to understand how there is formation of acyl CoA. Okay, now why this is important you will understand in 2 minutes, but I want you to write or draw this diagram of formation of acyl CoA. So usually this is a fatty acid. Let's draw with a different color. So usually this is a fatty acid, and this fatty acid will eventually get converted into a compound called as your acyl CoA, and there is an enzyme responsible here which converts this fatty acid into acyl CoA. And what is the enzyme responsible here? The enzyme is your acyl CoA, or we can simply call it as your acyl synthetase. So once again, acyl synthetase can convert the fatty acids into acyl CoA. Now, why the formation of acyl CoA is very important? Because I already told you that glycerol 3 phosphate will eventually get converted into phosphatidate, and during this step we have to convert, or we have to donate the acyl group from the acyl CoA. Okay, we are going to donate the acyl group. We are going to donate the acyl group to the glycerol 3 phosphate, and during this step the CoA will come out as the byproduct. So whenever the glycerol 3 phosphate needs to be converted into phosphatidate, we need acyl CoA, and glycerol 3 phosphate will accept the acyl group, and whenever the acyl group is accepted by the glycerol 3 phosphate, the CoA comes out as the byproduct. Okay, now how this glycerol 3 phosphate is converted into phosphatidate, we have to understand. So please keep the heading synthesis of triacylglycerols. Okay, now we are going a little bit deeper, so please try to concentrate here. As I already told you, we have glycerol here, and glycerol is a 3 carbon structure. To this 3 carbon structure, I am going to do phosphorylation at carbon number 3. So what will happen? It will get converted into glycerol 3 phosphate. So here is the glycerol which was having 3 carbons, and to the 3rd carbon I am going to add a phosphate group, and this compound is called as your glycerol 3 phosphate. What was the enzyme responsible here which was doing phosphorylation of glycerol? The enzyme is glycerol kinase. Okay, and this glycerol 3 phosphate, I already told in my previous video that some of the, this is the glycerol kinase. As I already told you in my previous video that glycerol kinase is absent in muscles and adipose tissue. So there the pathway is a little bit different. So there in the muscles and the adipose tissue we have dihydroacetone phosphate, and this dihydroacetone phosphate can get converted into glycerol 3 phosphate, and the enzyme responsible here is your glycerol 3 phosphate dehydrogenase. Okay, and once the glycerol 3 phosphate is formed, it will undergo acylation. What it will do? It will undergo acylation, and during acylation, acyl CoA gets converted into CoA, and what is getting added to the glycerol 3 phosphate? There is transfer of acyl group. There is transfer of acyl group to the glycerol 3 phosphate. So once again I am going to draw the structure of glycerol. So this was 1, 2, 3. What was the 3 carbon structure? It was glycerol, and to the 3rd carbon there was a phosphate attached which was glycerol 3 phosphate, and now we are going to add an acyl group to the 1st carbon. I will name it as A. Okay, so to the 1st carbon this is first carbon, this is the second carbon, and this is the third carbon. On the third carbon we have the phosphate, and on the first carbon we are going to add the acyl group. And what is the newly formed compound name as? The newly formed compound is named as 1-acylglycerol 3-phosphate, because on the glycerol there is one acyl group transferred, and on the third carbon we have the phosphate. Okay, hence the newly formed compound is your 1-acylglycerol 3-phosphate. And what is the enzyme responsible here, which is transferring the acyl group to the glycerol 3-phosphate? The enzyme name is your glycerol 3-phosphate acyl transferase. Okay, so in simple words, glycerol 3-phosphate is getting converted into 1-acylglycerol 3-phosphate, and the enzyme which is doing this conversion is your glycerol 3-phosphate acyl transferase. What it is doing? It is transferring the acyl group to the glycerol 3-phosphate by enzymatic activity. Okay, once 1-acylglycerol 3-phosphate is formed, eventually there will be second acylation, and during this step also what will happen? There will be conversion of acyl CoA into CoA. Okay, and once again there will be transfer of acyl group. There will be transfer of acyl group to the 1-acylglycerol 3-phosphate. So what will happen here? This is the glycerol which was having 1, 2, 3. On the third carbon we already have the phosphate. I will draw a little bit bigger here. First carbon, second carbon, third carbon, 1, 2, 3. I already told you that on the third carbon we were having a phosphate, and not only that, we were having an acyl group present on the first carbon. Now what we are going, there was a second acylation going on, and during this step there was addition of acyl group to the second carbon. So what is the new compound name? The new compound name is let's draw this with a different color here. The new compound name is 1,2-diacyl. Okay, diacyl, because two acyl groups are added to carbon number 1 and 2, hence it is called as your 1,2-diacylglycerol. Diacylglycerol. And on the third carbon we have the phosphate, so we will call it as your 1,2-diacylglycerol 3-phosphate. Okay, so 1,2-diacylglycerol 3-phosphate, the other name of this 1,2-diacylglycerol 3-phosphate is your phosphatidate. Okay, so from this phosphatidate, which is a common precursor for phospholipid synthesis and triacylglycerol synthesis, as I talked in my previous video. Once this phosphatidate is formed, we can synthesize your triacylglycerol and phospholipids. Okay, now what is the enzyme here that is responsible? What is the enzyme responsible? Enzyme here responsible for transferring this acyl CoA to the 1-acylglycerol 3-phosphate. The answer is very simple. 1-acylglycerol 3-phosphate acyl transferase, or you can simply call it as 1-acylglycerol 3-phosphate transferase. Okay, so first we were doing first we were doing first acylation was here, and the second acylation is here. So we are doing two acylations in the process, and the final product is your phosphatidate. Now I am going to draw the structure of phosphatidate here. What was the name of phosphatidate? First we will draw the diagram here. First carbon, second carbon, third carbon. 1, 2, 3. On the first carbon we have one acyl group, and the second carbon we have the one acyl group, and on the third carbon we have the phosphate. And what is the name of this one? Diacyl glycerol 3-phosphate, which was also known as your phosphatidate. Okay, so from this phosphatidate, I already told you we have to synthesize the triacylglycerol. Okay, so what we are going to do, we are going to do a little bit of cost cutting. Now who is the cost cutter here? There is an enzyme, and this enzyme will remove the phosphate group from the third carbon. Can you tell the name of this enzyme here? The enzyme name is very simple. It is your phosphatidate phosphatase. Okay, phosphatidate phosphatase, or PAP, is going to remove the phosphate on the third carbon of 1,2-diacylglycerol or phosphatidate. So what will be the final product? Once again, this is the first carbon, this is the second carbon, and this is the third carbon. 1, 2, 3. On the first carbon we have the acyl group, on the second carbon we have the acyl group, and on the third carbon we were having phosphate group, but this phosphate was removed. So what is the newly formed compound? The newly formed compound is your 1,2-diacylglycerol. Okay, so phosphatidate phosphatase is removing the phosphate present on the third carbon, and the newly formed compound is your 1,2-diacylglycerol. I hope you are clear with this. The other name of this one is also known as your diacylglycerol, and I already told you from diacylglycerols we can synthesize your triacylglycerol. So we are coming to the final pathway of the triacylglycerol synthesis pathway. So diacylglycerol needs to get converted into triacylglycerol. So what we are going to do, once again we are going to do acylation here. So once again here, acyl CoA will be converted into CoA, and during this step acyl group is added to the 1,2-diacylglycerol. And what is the enzyme responsible here? The enzyme responsible here is your diacylglycerol acyl transferase. Okay, so diacylglycerol will transfer the acyl group to the diacylglycerol. Okay, and what is the final product? Here is the first carbon, second carbon, third carbon, one acyl group, 1,2-diacylglycerol, and one acyl was transferred. Okay, so finally we have the three acyl groups transferred to the glycerol, and the newly formed compound is your triacylglycerol. And I want to talk the most important thing here is this diacylglycerol acyl transferase is considered as the rate limiting step. Okay, I know you guys are a little bit confused, but please watch this videos 2 or 3 times, then you will understand what is happening here exactly, or else you will not understand a single thing here. And I'm not only that, I will give a little bit of summary here, so please put summary. So I already told you glycerol can be converted into glycerol 3-phosphate, and what was the enzyme responsible? It was your glycerol kinase. And in the muscles and the adipose tissues, dihydroacetone phosphate was converted into glycerol 3-phosphate by the enzyme glycerol 3-phosphate dehydrogenase. Eventually, glycerol 3-phosphate needs to get converted into phosphatidate, and phosphatidate we are going to synthesize diacylglycerol, and from diacylglycerol we are going to synthesize triacylglycerol. Okay, first we need to do one acylation. The first acylation we are going to see. So here there is acylation. Acyl CoA is converted into CoA, and during this step acyl group was transferred. And what is the enzyme responsible here? The enzyme responsible was your glucose 3-phosphate acyl transferase. Okay, and what is the newly formed compound here? It was your let's draw a different color here. The newly formed compound is your 1-acylglycerol 3-phosphate, because on the first carbon we are going to add the first acylation acyl group. Okay, eventually we are going to do the second acylation, and during this step acyl CoA gets converted into CoA, and the acyl group is added. And what is the enzyme responsible here? Which is your 1-acylglycerol 3-phosphate acyl transferase. Okay, and finally, so what is the newly synthesized compound here? It is your 1,2-diacylglycerol 3-phosphate, which is also known as your phosphatidate. And from this 1,2-diacylglycerol 3-phosphate, aka your phosphatidate, we are going to remove the phosphate. And what was the enzyme which was cutting off the phosphate? It was your phosphatidate phosphatase. And what was the compound formed here? It is 1,2-diacylglycerol. And once again the final step was addition of acyl CoA, and from acyl CoA was formed, and we were transferring the acyl group to the 1,2-diacylglycerol. And the important enzyme here is your 1,2-diacylglycerol acyl transferase, or you can simply call it as your diacylglycerol acyl transferase. Okay, and the final product is your triacylglycerol. So this is how it was written in your biochemistry textbook. I know this is a lot tough than any of the biosynthesis mentioned in your chapter 24, but trust me, if you watch this video again you will understand everything. I would suggest watch it 2-3 times so you will be clear with this. Now as you can see here, this was the pathway that was given in your chapter 24. So as you can see here, glycerol and dihydroacetone phosphate will get converted into glycerol 3 phosphate. Whenever glycerol is getting converted into glycerol 3 phosphate, the enzyme responsible is your glycerol kinase. Whereas in the case of dihydroacetone phosphate, it is glycerol 3 phosphate dehydrogenase which will convert the dihydroacetone phosphate into glycerol 3 phosphate. And as you can see here, there will be a first acylation here where the acyl CoA will donate the acyl group and get converted into CoA, and the enzyme responsible here is your glycerol 3 phosphate acyl transferase, and this will transfer the acyl group to the first carbon. Hence there will be one acyl group present on the first carbon and the phosphate present on the third carbon. Hence it is called as your 1-acylglycerol 3 phosphate. Okay, and eventually 1-acylglycerol 3 phosphate will undergo second acylation, and during which a second acyl group is added to the second carbon of the glycerol. So eventually it will get converted into 1,2-diacylglycerol, because on carbon number 1 and 2 there will be presence of two acyl groups. Hence it is called as your 1,2-diacylglycerol, and on the third carbon we have the phosphate. Hence it is called as your 1,2-diacylglycerol 3 phosphate. And the other name of 1,2-diacylglycerol 3 phosphate is your phosphatidate. Okay, so from this phosphatidate we are going to remove one phosphate. And what was the enzyme that is cutting down the phosphate at the third carbon, which was your phosphatidate phosphatase, or the other name is also known as your phosphatidate phosphohydrolase, but in your textbook you just remember that one as your phosphatidate phosphatase. Okay, which will remove the phosphate at third carbon, and the only thing that is left over is your acyl groups at carbon number 1 and 2, which is known as your 1,2-diacylglycerol or diacylglycerol. Eventually to this 1,2-diacylglycerol we are going to donate acyl group again by the enzyme diacylglycerol acyl transferase, and there will be formation of your triacylglycerol. So that was about it, guys. I know you guys are a lot confused, but watch this video 2-3 times, then you will understand this one clearly. Thank you, guys.